Namaskar दोस्तों, welcome to channel Toxicology on Board. In this video, we will be learning about topic fluorescence in situ hybridization fish technique. And the last video we were talking about northern blotting technique. So these two video questions are related to the type of assays which are used to identify certain kind of damage. So let's see what fish is there. What is the question with respect to this particular concept? So fluorescent conjugated nucleic acid probes are used as tools in techniques called fluorescence in situ hybridization. So in this FISH technique, what we are using? We are using fluorescence conjugated nucleic acid probes. For what primary purpose is the technique used in genetic toxicology? As I said, no, these assays to identify what kind of either target organ damage or non-target organ damage. So this question is related to non-target organ, that is the non-target organ damage with the genetic toxicology. So let's see what options we have. Okay, so we have option number A, facilitates counting micronuclei in polychromatic erythrocytes. Option B, locates point mutations in mammalian chromosomes. Option C, more accurately quantitates sister chromatid exchanges. Option D, facilitates cytogenic analysis by chromosome painting. So the right answer is option D. What is option D? Facilitates cytogenetic analysis by chromosome painting. So this question is taken from the series of American Board of Toxicology. The explanation given by ABT is FISH. FISH is fluorescence in situ hybridization, uses fluorescent probes to bind and visualize nucleic acid. Okay, to bind and visualize nucleic acid with a high degree of similarity. So if you want to learn in detail more, this is a reference which is given by ABT. Please go back and read Clayson textbook 8th edition, page number 465 and 466, and page number 471 and 472. So let's see what we can learn from this concept. So a FISH assay refers to fluorescence in situ hybridization test, a laboratory technique that uses fluorescent probes to detect specific DNA sequences within cell or tissue. If you see, my last video was about RNA detection. Okay, that was northern blotting technique. Now, FISH assay is for DNA sequences within the cells or tissue, and in that question it was tissue sampling. The next, these probes bind to complementary genetic material, allowing scientists to visualize and map genes, identify genetic abnormalities. Like what are those abnormalities? Deletions, duplications, translocations, diagnose genetic disorders, including some cancers and prenatal conditions. So all these type of damages related is map genes can be done with the help of this FISH assay. Okay, the technique is used in research and clinical setting to study chromosomal structure. Okay, if you look at the chromosome structure, we have centromere. Okay, then if you telomere, all this, if you remember the typical chromosome structure, na, so it will be this FISH assay will be used to study the chromosomal structure and gene location, providing insights into genetic disease and treatment responses. Okay, so how this fish works is we have three steps. One is probe preparation. First you have to prepare the probe. Second one is hybridization. The third one is visualization. Coming to application of this fish technique, we have genetic disorder diagnosis. It can be used, fish technique can be used for cancer detection. Fish technique can also be used for prenatal testing. As you can see, prenatal conditions it can be used, and it also used for our research purposes also. So what are the advantages of fish technique is it is used for spatial information, because here you can see, no, because of this hybridization, you can see spatial information of the genes can be seen on the chromosome versatility. So this assay, as you can see, it has got various applications. It can be used for various types of genetic damage like deletions, duplications, translocation, etc. And it is also used as a diagnostic tool to identify certain genetic type of diseases also. Which technique can be used. So as I said, no, there are few topics in the case of DABT syllabus. If you see, they'll be asking about the type of the assays and the assays which they are used to identify what kind of damage, either it is target organ damage or non-target organ damage. So in this 50 series of questions, one, you people can read about the topic Northern blotting. Second, you people can also learn about the fish technique. Okay. Mm, so thank you so much. Be curious. For any questions, you can always write a mail to arya.priyanka18@gmail.com. Like, subscribe, and follow my YouTube channel, Toxicology on Board. All the best for your exam. Thank you.